Hello, I'm Mason Brooks, Managing Editor of Epigenie, and welcome to today's webinar presented by Dr. Bryce Alves titled Precision Mapping, High-Resolution Mapping Using ChIP-Exo. Dr. Alves is a research scientist at Active Motif, where he and his colleagues have worked really hard to optimize and streamline the ChIP-Exo method so that it's even more robust and accessible than ever. And that's what he'll be discussing more with us today. Before I hand things over to Dr. Alves, I'd like to thank Active Motif for their support and sponsorship of this webinar. Active Motif is committed to enabling researchers who investigate chromatin biology and epigenetics through their development of high-quality products. These include chromatin IP kits and reagents, high-quality antibodies to histones and chromatin proteins, and kits to analyze DNA damage, DNA methylation, and histone modifications. So please check out the Active Motif website when you get a chance. One last note before we begin. We'll have a question and answer session after the presentation, so please type any questions that you have in the questions box. This will appear on the right-hand side of your screen, and I'll be sure to pose them after the talk. So with that bit of business taken care of, I'll go ahead and hand things on over to Dr. Alves.
Thank you, Mason. I would like to thank Epigenie for the invite to present the study on ChIP-Exo, and hopefully with all those that are intending to listen to this talk and who will later view this talk, I will greatly inform you on how ChIP-Exo works, and maybe then more of you will use ChIP-Exo in the future to carry out your sequencing needs. Okay, what is ChIP-Exo and why should you do it? ChIP-Exo has advantages over what are you standardly using for sequencing in standard ChIP-Seq. And these advantages that are apparent in ChIP-Exo address many of the current limitations you see in ChIP-Seq, such that ChIP-Exo is able to identify more concisely defined binding sites by increasing the resolution of which you see these binding sites. In addition, because of its accuracy in aligning and developing peaks, you have reduced background reads. And because of this, you actually require less reads to get adequate coverage to identify binding sites using this sequencing technology. So this talk will cover ChIP-Exo as it relates to ChIP-Seq historically, with a more in-depth look at the ChIP-Exo protocol to give you a better understanding of just what's happening in the ChIP-Exo reactions. And there will be then a quick overview of data analysis at the end of that section. This will be followed with ChIP-Exo data that we have generated here using the kit that we've recently developed at Active Motif versus that which has been publicly available and published in the PubMed forums. And in addition, we'll also have a comparison of ChIP-Exo data to ChIP-Seq data and how it relates and how it differs. And at the end, as Mason said, we'll have a question and answer session. So with ChIP-Exo, when we first started looking at protein-DNA interactions, the real first technology that we had to identify where a transcription factor interacted with DNA was really the ChIP-ChIP technology. And here you had a big fragmented library of DNA with your transcription factor of interest. You then ran this across a microarray containing oligos for the whole genome, and what you would get was an identification of where the protein was bound, and it would result in basically a very large peak with a resolution of about 500 to 1,000 base pairs, which wasn't really an identification of the sequence per se where your protein or transcription factor actually interacted with the DNA, but it gave you a general idea of where that sequence might be found. This technology was replaced by the ChIP-Seq technology, which was a leap forward in the ability to identify transcription factors binding to DNA. Here you have your chromatin or DNA, which you cross-link to proteins or transcription factors, either way with formaldehyde or other forms of cross-linking, and then carry out fragmentation using sonication or enzymatic fragmentation, and this generated a large amount of fragments as we see here. You then use enzymatic reactions to clean these fragments up. You can then add an adapter sequence onto either end of these fragments by ligation, and from these ligation of adapters you can then use a indexing primer set, which allowed you to add multiple barcodes throughout multiple samples, as well as allow you to then run these samples out on one of the sequencers that came out, such as a HiSeq or one of the NextSeqs out there. And this allowed you to actually sequence the DNA, and it gave you a resolution of binding for where your protein has interacted with the DNA of about a resolution of 100 to 400 base pairs. And as we see in the example data down here, you have much tighter peaks, and this gave you a more focused idea of where the transcription factor actually was interacting with the DNA, and from this you could actually generate sequences and motifs that were identified to be associated with certain transcription factors. And this was the prevailing technology for several years until Frank Pugh at Penn State and his lab came up with the brilliant idea to use exonucleases to trim back the DNA fragments that you generate during fragmentation to get a much tighter resolution and identification of where a transcription factor is binding to the DNA. And this occurred in about 2010 with two publications that came out following their discovery of this technique in 2011 and 2012, and these were based on solid background for sequencing. And that was the initial steps into ChIP-Exo. Later, Jason Carroll lab in Britain actually was able to adapt this ChIP-Exo technology to work with the Illumina platform, and also Frank Pugh's lab very shortly after that published their own paper where they adapted this technology to the Illumina platform, making ChIP-Exo accessible on two different platforms and more accessible for use in many of the sequencing technologies out there. So how does ChIP-Exo work? Well, it starts out much the same way as you start out a standard ChIP-Seq reaction to get to sequencing. You have your DNA, which you cross-link to your transcription factor of interest, and you then carried out fragmentation, and in this case we're using sonication to generate fragments of differing lengths. You then, just like in ChIP-Seq, would like to generate blunt-ended fragments for ligation, so you clean these up using a cocktail of 3 enzymatic enzymes that run concurrently with each other: plenal polymerase, T4 polymerase, and T4 polynucleotide kinase. And these are able to clean up your fragments to not have overhangs, but actually have blunt ends on each side, which makes for the easy addition of an adapter sequence. In ChIP-Exo, this is where you add your first adapter set, and these adapters are special and unique to ChIP-Exo in that on the 5' end of these adapters, you have a phospho group on both set of adapters, and this is very important for the initiation of exonuclease activity by lambda exonuclease, which requires a phospho group to be on the 5' end. So we can add these adapters much the same way as you would add your adapters in ChIP-Seq, using T4 ligase to, in this case, blunt end ligate to our transcription factor containing DNA fragment. And now we have our adapters ligated. We actually want to clean up our DNA further, and that is to do some nick repairs that may have occurred during our sonication that were not originally taken care of, as well as we want to take care of the overhangs that we have from our adapter ligation, as we see here. And the enzyme that is used in ChIP-Exo to carry this out is to use Phi29, which has the ability to do nick repairs as well as blunt end any fragments that exist, which we need to do. So this fills in any nicks and blunt ends it, and now you have a fragment that is actually perfectly ready for the exonuclease activity of lambda exonuclease. And so, as I stated before, lambda exonuclease requires that there be a blunt-ended, double-stranded DNA which has a phospho group at the 5' end, or it will not initiate its exonuclease activity. But we've added these to our fragment, as you can see here. So these lambda exonucleases recognize the 5' blunt-ended DNA and begin their exonuclease activity, running 5' to 3', removing the nucleotides one by one, until they come to the point where it runs into a region where we have a transcription factor that is cross-linked. And this transcription factor is actually acting as protection for the DNA underneath, since this blocks any more exonuclease activity to occur and protects the binding site for the transcription factor. So we've now shortened each fragment on each strand of the fragment to a much shorter size, as well as we have isolated where the transcription factor is binding to the ends of these fragments. So what we want to do now is we do a standard reverse cross-linking using proteinase K to remove the cross-linked transcription factor, and then high heat to separate the fragments. Now these fragments still contain the original adapter sequence that was found on the 3' end, and this allows you to add a primer, which can then be attached to these the remaining adapter sequence, and using once again Phi-29 as a polymerase, we're able to generate double-stranded DNA once again from these single-stranded fragments. And now using these double-stranded DNA fragments, we see that we have 2 fragments, once again, which are smaller than the original fragment that we originally had. And we now come in with a second set of adapters, and these adapters we ligate onto these fragments using, once again, T4 ligase reaction, and it is these adapter sequences which will match up well with your indexing primers, which allows you then to use your indexing primers to create a small level of amplification. Usually our amplifications are about 12 to 14 cycles, and you then do a size selection on a gel to clean up your DNA fragments that you've generated and have indexed, and to get away from any free-floating indexing primers or adapters that are still floating in the mix. And then you are ready to proceed off onto sequencing like you would with ChIP-Seq. And to give you an actual pictorial reference of what ChIP-Exo is exactly doing, you can actually see ChIP-Exo and its function by doing a walking primer reaction using PCR and then running these out on an agarose gel. So what you do here is you have your DNA fragment and an identified binding site for the transcription factor of interest. You can determine this from the ENCODE databases if it's known, or maybe from your own experiments with ChIP-Seq. And you can design primers to the 5' running 5' to 3' being your universal primer, and you have reverse primers that you then walk out from the initial known binding site between these primers at 200 base pairs, 300 base pairs, and 400 base pairs away. And then you can compare this with ChIP data. So if you just perform ChIP with no ChIP-Exo, you see this very nice stair-step product that you get on your agarose gel out to about 380 base pairs in this case. Now, when we look at the ChIP-Exo treated DNA, we see that it has been trimmed back in that you still see the initial 89 base pair fragment or DNA product that you get from PCR, as well as the next 2 steps occur, but as further on than that, there is actually no product being really produced. And that's because the exonuclease has now trimmed the DNA fragment back to where there is no landing site for these primers. And because there is no landing site, you won't get a product, and that's exactly what ChIP-Exo is doing, in that it is generating smaller fragments that are more concise and to the point of protection by the transcription factor. So when we go back to our original slide, which we're comparing the different technologies out there, ChIP-Chip, ChIP-Seq, when we get to ChIP-Exo, you have really reduced the amount of reads that are going to happen down to a very concise region of buildup just before the protection point, which we see here. This buildup, and this is where your transcription factor has been bound, protecting it from digestion, and you see it on both the forward and the reverse strand. And the space between these is, of course, where the transcription factor binding site is. And because of this very concise buildup of these reads at this very small point, you get a very tight resolution for the identification of a binding site, which we see here, with a resolution that can range from 14 base pairs up to 100 base pairs, which is a significant improvement over just simple ChIP-Seq, where the best you're going to get is about 100 base pair resolution. So when you generate ChIP-Exo data, it's actually treated differently at the end analysis than you would with just traditional ChIP-Seq. Mapping to the genome is very similar to ChIP-Seq. However, you don't want to remove your duplicates. In ChIP-Seq, you often remove all your duplicates because you don't want duplicate reads. You want unique reads aligning to the genome. In ChIP-Exo, that's not exactly how the biology works. Since you are trimming your fragments back to a very concise area, you are going to inherently, on its own and naturally through the biology, generate lots of duplicate reads, which you want to retain because you want to actually build up these reads at the point of where the transcription factor has protected the DNA, and you want to see a very large spike, or what is known as a border, occur, and then a drop off from this. And you're going to see this in the next slide when I present this. Now, the programs that are out there that you can use to look at ChIP-Exo data, many of us have used MAX in the past to look at our ChIP-Seq data. And MAX also can be used to look at ChIP-Exo data, although on the standard setting of MAX, you're going to see reduction in the peak size, but not to the point that I've been describing at about 100 base pairs down to 14. You're not going to see that with MAX unless you apply very unique filters to get to that point. And so you can't just use the default settings with MAX. You're going to have to work around with MAX to get it to look at ChIP-Exo data in the way that you can see the resolution. The original program designed to look at ChIP-Exo was developed by Istvan Albert, and that's GeneTrack. This was the program used at Penn State by the Frank Pugh Lab, and is still being used by Frank Pugh's lab to analyze their ChIP-Exo data. This program is more unique in the way that it looks at ChIP-Exo data than what MAX looks at ChIP-Exo data, in that it looks at the buildup of reads on both the forward strand and the reverse strand, and therefore it can identify the protected site between these buildup of reads, which is what you really want to look at. Another program, which is the program we use here at Active Motif that does this, was developed by Li Go Wang, and that's the MACE program. And this program uses several pairing algorithms to look at the buildup of reads on either the forward strand and the reverse strand, and using this buildup of reads and these pairing algorithms is able to identify the binding site between these borders. And you're going to see that in this next slide. This is what you see when you use MACE to analyze your ChIP-Exo data. And this is a graphical representation of what the bigwig files will look like. This is the forward strand, this is the reverse strand. So as I've been telling you, you want to see this buildup of reads right up to the border site. And this is what I'm talking about. You see this buildup of reads right to the border, which is this protective border where the transcription factor protected any DNA from getting digested. And then you see a drop-off with a very small bump representing the second peak or border that would be protecting it. Now when you look at the reverse strand, you see the exact same image just as a mirror image, where you see a buildup of reads to this border, and then a drop-off, and then the mirror image of the second border, which lines up with the forward strand. So MACE is designed to look at this buildup of reads at these different borders, and it identifies both a forward binding border on the forward strand and a reverse border on the reverse strand, and then uses its pairing algorithm to combine these borders and identify how these borders align with one another. And the end product is using the Gale-Sharpley mating equation, which mates the pairs between that and identifies the binding site between these as a GSB binding site. And this is the site where you identify the binding sequence of your transcription factor, since this is the site between the two peaks where it was protecting the DNA. And that's the program we use here, but as you can see, there are other programs that can be used to look at ChIP-Exo data. Now, Active Motif recently, as of last Friday, released its own ChIP-Exo kit. And this kit was optimized to use a much shorter protocol to reduce the amount of hands-on time for your ChIP-Exo reaction. The kit contains 2 complete modules, which will allow you to go from cells growing in your plate all the way through the fragmentation, ChIP-Exo reaction, and the generation of a sequencing library, which you can then send off to either a core facility for sequencing, or maybe you have a sequencer in your lab, and you can carry out your sequencing there. Using our kit, as you'll see in the coming results, we're able to get results from as little as 20 million cells. Our kit, we find that the data has significantly reduced background over that which we see with traditional ChIP-Seq, which allows us to identify much more concise binding sites for the transcription factors, as well as we're seeing that more and more novel binding sites that we had not seen in our ChIP-Seq data actually come up using ChIP-Exo. So here's the first piece of data that we have. This data was generated to CTCF, and this is the bigwig files you get from using the MACE program, which I previously showed you a nice idealized version that I had generated by hand. This is what they actually look like. The top panel is published ChIP-Exo data for CTCF at a certain site, and using 60 million cells, they were able to identify this binding site to have 45 base pairs. Active Motif, using our kit, using 20 million cells, identified the exact same binding site for CTCF, but with a base pair of 46 base pairs, so we're one base pair off. When we compare that to our traditional ChIP-Seq data that we have here, using 60 million cells, we found that the binding site identified for ChIP-Seq was 96 base pairs. So essentially, using the ChIP-Exo technology, we're able to have the size of the binding site that's identified. And when you turn on the ENCODE database, if you're using Genome Browser and run the ENCODE database, this exact same binding site for CTCF is identified to be 119 base pairs. So as you can see, ChIP-Exo is better than, is a better, much better resolution by about 2 times that what you see with the ENCODE database. Data generated from this ChIP-Exo experiment were run through Homer to identify exactly what binding sites we were identifying using ChIP-Exo, and the top 2 binding sites were both CTCF binding sites, as you can see here. Another data set that was publicly available that we decided we would like to look into was that for FOXA1. As we see here, the published data set for FOXA1 used 100 million cells and identified the FOXA1 binding site to have a size of 70 base pairs, whereas Active Motif, using 20 million cells, once again, which is our standard number, we identified the same binding site to be as low as 14 base pairs. And when we compare this to the ENCODE database, this binding site in ENCODE has been identified to have a binding site of 215. So in regards to FOXA1, you can definitely see that ChIP-Exo significantly reduces the amount of base pairs in your binding site compared to that of traditional ChIP-Seq known data. And once again, these samples were run through the Homer program looking for which motifs were being bound, and we find that both FOXA1 and FOXA2 were the predominant binding sites that were being found, which is very nice, which you expect to see. Now, many of you probably watching this are saying, well, that's very well and good that you can get such data using CTCF and FOXA1, but those transcription factor binding sites are some of the most ubiquitous binding sites throughout the genome. What really happens when you look at a less pronounced transcription factor binding site? And we had that exact same question. And here we're in collaboration with the Alexander Hoffman lab at UCLA, where they asked us to look at mouse embryonic fibroblasts, which were either not stimulated or stimulated with TNF-alpha, and they wanted to know what ChIP-Exo would say about the binding sites that they're identifying. And they provided us with an antibody, which they said did work pretty well in ChIP-Seq, but they hadn't really had the greatest success with this antibody. So using that antibody in our ChIP-Exo reaction, when we looked at the TNF-stimulated samples, you see that we identify a very tight binding site right here at the end of the TNF gene, whereas when we compare it to the ChIP-Seq data that was provided to us by the Alexander Hoffman lab, we see that TNF, when we looked at their traditional ChIP-Seq, has a large amount of reads covering all the way across the gene. With any of these could be a binding site, but we do see that this binding site down here, which we also identify down here, is much wider and much larger than what is identified using the ChIP-Exo technology. A more prominent example of what I'm talking about is what you see definitely when you look at NF-kappa B inhibitor alpha gene. Once again, stimulated with TNF, you see a very concise binding site being identified by ChIP-Exo, whereas when you look at traditional ChIP-Seq data from the Alexander Hoffman lab using TNF-stimulated MEFs, we see a very large binding site being identified, which starts all the way out in the promoter region and goes 1/3 of the way into the NF-kappa B gene as being identified as the binding site. You can definitely see from these two examples that ChIP-Exo definitely produces a very concise identification of the binding site for REL-A in this case. Well, I've been talking to you about identifying many of the similar binding sites that you would expect to see. You also see in ChIP-Exo that you can find de novo sequences for binding that have not been identified using traditional ChIP-Seq. And a good example of that is in an experiment we ran here several times using ChIP-Seq for P53 as well as ChIP-Exo for P53. And then we compared these. In our traditional ChIP-Seq for P53, binding on the P21 gene, which is known to be highly regulated by P53, you see two historically identified binding sites, one at the promoter region and one just before the second exon. When we use ChIP-Exo, we identified 4 binding sites repeatedly. The initial binding site we see here at the start, this matches up perfectly with the ChIP-Seq binding site, and we also identify this binding site, which is probably the same as this one. That's probably just due to differences in the resolution of the ChIP-Seq compared to the ChIP-Exo, but we also identify 2 novel binding sites within the P21 gene for P53 here and here, which indicates that one can use ChIP-Exo not only to look at the binding sites that are already known and getting more concise identification of those binding sites, but you can also find binding sites that were previously unknown to your previous ChIP-Seq data. So this is a representation of the number of peaks that we usually see and the size of the peaks. The top panel here is analysis of ChIP-Exo data looking at using MAX, and the bottom is MACE, and this was just to give you an idea of the difference between MAX and MACE, as I alluded to earlier when saying that you can use MACE, but you're not going to get the same amount of resolution. When you use MAX, you see that the binding sites that are identified using MAX, even when you have ChIP-Exo, are rather large. 141 here, the published data is with 94, compared to traditional ChIP-Seq, which is 297. So you're actually reducing it by at least half if you take the average of these. And that's the same as seen for CTCF. Yet if you use the ChIP-Exo specific program, such as MACE, as we see here, you're able to identify using this algorithm that looks more at the border buildups and the protection site. You see that the identified peak length is about 33 base pairs, 38, which is much improved over that which you would see in traditional ChIP-Seq. As we see here, you also see it in CTCF. You'll also probably notice that there are some differences in the amount of peaks called, and that's because MACE is very restrictive. And here we're actually only looking at the GSB border pairs. MACE actually identifies a greater amount of peaks, but if it cannot mate these peaks to a forward and reverse, it only gives you a single forward or a single reverse peak, and I have excluded those. So you actually get very comparable numbers to what you see in MAX, but these are the perfect border pairs, which means it's a very accurate binding site that you're identifying. And as you can see, with higher numbers of cells, you actually are able to identify more binding sites, whereas we only use 20 million cells here in our experiment. In 100 million cells, you actually about doubled the amount of binding sites that are identified using MACE. So that's just a warning to say that you can get data with 20 million cells, but if you use more data or more cells, you're going to actually get more binding sites that are identified. So with that, this is the points that I'd like you to take home today, which is ChIP-Exo data combined with ChIP-Exo specific analysis programs and software will increase your resolution over traditional ChIP-Seq, and I believe I've shown you that. You identify binding sites with a resolution of binding for about 20 base pairs up to 95 base pairs on average for your binding site, compared to the traditional 200 base pairs you observe with just ChIP-Seq. You can get, with using the ChIP-Exo kit from Active Motif, results from as little as 20 million cells, but if you use higher amounts of cells, you're going to identify far more peaks. The ChIP-Exo kit provided by Active Motif enables you to identify not only better resolution binding sites, but as you also can identify unique transcription factor binding sites that may have been missed by traditional ChIP-Seq. And the kit that is provided here can go from your cells in a plate all the way to a sequence-ready library in a single assay. And for acknowledgments, I would like to acknowledge, of course, Frank Pugh, who came up with the technology for ChIP-Exo at Penn State. I'd like to also acknowledge Alexander Hoffman at UCLA and his graduate student Kim for the provision of the mouse embryonic fibroblast, which we then carried out the REL-A ChIP-Exo data that I showed you. And I would like to acknowledge that Ligo Wang, who is now at the Mayo Clinic, who developed the MACE software, which we use predominantly here to analyze our ChIP-Exo data. We find it to be very excellent software for this, although many softwares are now becoming available for ChIP-Exo, each with their own individual abilities and strengths and weaknesses, and end users should evaluate the software based on their needs. And from that, I'd like to end this talk and take questions.
Great. Thanks, Dr. Alves, for that really great talk. And I will now open up the Q&A session, so please submit any questions you might have if you haven't already. If we happen to run long and your questions aren't addressed, don't worry. I'll make sure to pass them along to Dr. Alves and the Active Motif crew for some follow-up later. There have already been a few posed, so we'll get right into it. These first three were early on in the talk and sort of related. So first off, for a regular ChIP-Seq, you often need to sequence input samples. Would this be required for ChIP-Exo as well?
We have not seen that you need to sequence input samples. We've done some input samples, but with the ChIP-Exo technology, due to the amount of manipulation, basically, of the sample as you run to the next one, it gets so different from what you initially put in because you're cutting it back, making it more concise, that a comparison between an input with that of a ChIP-Exo sample is like comparing apples and oranges. That's what we've found it to be. I hope that answers the question.
Right. And sort of related, you also normally, with normal ChIP-Seq, need to confirm by ChIP qPCR. Is this needed for ChIP-Exo as well? Well, I'll let you answer yes or no on that.
Yeah, we have actually gone back and done that. So we usually end up running the ChIP-Exo samples and sequencing them, and then to confirm that that is in fact a binding site, we have carried out qPCR to look at that binding site. We've of course used the ChIP-Exo data to design primers and then gone back. But another way you can also look at it just with standard PCR by using the walking primers if you'd like to see if it's protecting it or not.
Okay. So would those need to be prepared alongside with like the exonuclease digestion and adapter ligation as well, if you were gonna?
You get so much library from 20 million cells. You could actually and if you're using qPCR, which I assume you'd be looking at to identify these sites, you can simply save a small aliquot, a diluted small aliquot that then can be used after your ChIP-Exo if you're interested in confirming for that exact same experiment, or you could run a separate sample We often just separate a small sample, which we dilute about 1 to 20, 1 to 30, and then we can pull from this, since we're going to use it with qPCR, and we use our primers in qPCR we can confirm that this is in fact a binding site that we've identified.
Okay. So this next question was referring to sort of steps around the area of steps 3 through 5. And the question was just, is the TF still there at this point? And you went on to explain that it was. So maybe you could answer when the transcription factor actually is removed from the DNA.
So yes, so you keep the transcription factor on all the way to the point where you have now digested everything off of the DNA, and with the the lambda exonuclease, where it ran into the protected region by the transcription factor. There's a reverse cross-linking step, and here you do proteinase K for about 2 hours, and that'll of course digest any protein left in the reaction. And from that you can then do a separation of the overlapped DNA that still remains by high heat. So that's what you're doing there.
Okay. This next question is referring to the data analysis steps. So without removing duplicates, is there a recommended QC for library complexity or over amplification? Or is that an issue at all? Or?
That's a good question. We definitely have noticed that, well, let me think here, what we've looked at. So we always leave our duplicates in. If you remove them, we've done it once where we removed our duplicates and you get no signal, because that's exactly what the biology of ChIP-Exo is to do, is to build up reads. Library complexity, we usually are just simply looking at the unique aligning reads. So not per se duplicates, but at least reads that align, and we're looking to at least get about 20 million unique reads to align to the genome. But we really haven't seen a whole lot of, amplification, duplications of reads that you would see with a high duplication when you do indexing primer over amplification, since we keep our number of amplifications down very low, and we keep our samples separate from each other to keep the amplifications very low. But that's more detailed in the kit. I didn't go into that depth of detail here in the talk.
Right.
Yeah.
Great. So what sequencing platforms is the Active Motif kit compatible with?
The Active Motif kit is made to work with Illumina. The adapters are all designed to work on the Illumina platform under ChIP-Seq. So Frank Pugh's lab has generated a large amount of their data on the solid platform using their own adapters. We don't have those adapters right now in our kit. It's more the Illumina-based adapters.
Okay. Would it be difficult for somebody to create adapters and sort of tweak your kit to work on another platform, or would that be difficult?
The one thing I would warn people about doing that is you have to make sure that your adapters will generate a double-stranded DNA that has a phospho group. That was one of the hardest parts about transferring this technology from what Frank Pugh did with his adapters to the Illumina set, in that Illumina adapters are designed to have a Y shape, but you really want double-stranded shape to occur. So if you're developing your own adapters, you would need to have be sure that you would have double-stranded. And if you want solid-based adapters, I would definitely say look at Frank Pugh's data and use his adapters.
That's a good tip. Thanks. So next step, or I'm sorry, the next question is: do you also include the RecJF, and I'm sorry, I'm not an expert on.
Yes.
I don't know what that is.
Yes,
the RecJF exo step in addition to.
Yes, I do. For simplicity of this talk, I left that step out just to keep the talk rolling along. But following the lambda exonuclease reaction, you then do a second digestion, and that's to use RecJF, which is a single-stranded exonuclease that will run 5' for 3', and this is to eliminate any single-stranded DNA that may be binding to your transcription factor of interest, and therefore it's like a double whammy whereas the lambda's getting rid of all the double-stranded running 5' to 3'. If you had some DNA that was single-stranded that was still attached to a transcription factor, you wouldn't want those reads to be messing up your sequencing because they'd be much longer and not digested. So you use the RecJF that then runs along 5' to 3' as an exonuclease until it hits the transcription factor. So just for simplicity of this talk, I didn't include that step, but we do in fact use that in our kit. It's the next step following the lambda, as someone pointed out.
Okay. So why does your kit use blunt end ligation as opposed to like a poly-A tailing technique?
We developed this kit to use blunt end ligation simply because we designed the primers, and the primer set that we finally got to work were actually designed to be blunt end primers or adapters rather than with the A tail. We could definitely convert ours to directional using A tailing, and we just have to add a T to the ends of our adapters. But since we want these adapters to simply land on either side of the blunt-ended DNA that we already have, it doesn't really matter if you have directional, since they're the exact same adapters, just in mirror image from one another on either side.
Okay. Do you think ChIP-Exo would be adequate for detecting a different histone modification?
Now that's a very interesting question, and this is actually what our collaborators at UCLA in the Alexander Hoffman lab have started to do with ChIP-Exo. I haven't heard the results from their studies yet, but they were so thrilled with how our data looked when looking at REL-A that they have a branch that's looking into histone modifications or phosphorylations and all that on the DNA. And so I haven't heard what their results have been, but they have been bouncing back ideas back and forth and asking questions, which tells me that they're beginning to work on that.
Mm-hmm. So it's at least sort of theoretically possible.
It's theoretically possible. I would definitely think that it will work, but I can't say that I've ever seen it work. So.
Mm-hmm. What's the lowest amount of input cells that you guys have been successful using ChIP-Exo with?
I have gotten very good coverage as low as 10 million cells, and I have actually gone as low as 4 million cells, but the library coverage and the peaks were very small when we only had 4 million cells to start with, so I would not recommend going below 10 million. 10 million would be my lowest, but for very good coverage, I would say stick with 20 million cells.
Okay, so not quite a low input solution at this time.
No, you definitely get reduced amount of reads aligning, and many of the ChIP-Exo programs that we looked at had a hard time identifying peaks because the buildup of reads were so small.
Right. All right, so this is a little bit of a long question, but I think pretty interesting. If you were to compare CTCF binding data from ChIP-Seq and ChIP-Exo, how many new binding peaks do you think you would find for CTCF, considering that it binds so many places across the genome? And same for P53.
Yeah, we have looked into both our data sets, both for CTCF and for FOXA1, comparing ChIP-Exo overlap of peaks with that of ChIP-Seq for those same transcription factors. For CTCF, we found that we had about 50% of the peaks were novel compared to that of ChIP-Seq, and when we looked at FOXA1, which is very ubiquitous, much like CTCF, we found an even larger proportion where about 70% of the peaks that were identified were novel compared to those that we traditionally had seen in FOXA1. So we definitely see a large amount of peaks that have not previously been identified by ChIP-Seq.
Mm-hmm. Oh, that's pretty powerful. So kind of a technical question here. The exonuclease digestion is normally 30 minutes, but since considering that's so critical to the whole protocol, would you suggest going longer or shorter? Or
so
maybe
we have done experiments where we've gone longer, as well as we've added more, 10 times the amount of the exonuclease to either, increase the speed of the reaction. And in both experiments, we did not see any significant difference from the simple, I think it's 5 units of lambda exonuclease for 30 minutes than what we saw with 50 units of exonuclease in 30 minutes or in an hour. It didn't seem to have any real significant effect on the resolution of your results.
Got it. So I was not sure if I understand this question. So do you know of any examples of ChIP-Exo being applied in bacterial systems and reasons why it may or may not be useful in prokaryotic organisms?
I don't think there's any published data for the bacterial systems. Now, Frank Pugh initially produced this assay looking at yeast, so on the yeast level you can get down to it, but I can't see why you wouldn't be able to do bacterial systems. It seems like it would, work right along with what you traditionally can do.
So what would a typical amount of antibody for ChIP-Exo with, say, 20 million cells be? Is it equivalent to standard ChIP-Seq stuff?
Yes, we use what we standardly use for our ChIP-Seq reactions, which is 10 micrograms of antibody. If its concentration of that antibody is known, if it's purified antibody and it's known that there's a concentration, you usually use about 10 micrograms of antibody per reaction for 50 million cells. If it's a polyclonal that is unknown, you're basically just going to have to guess because you don't know really the concentration of antibody binding there. So to hedge our bets, we usually use about 15 to 20 microliters out of that, assuming that it's probably polyclonal and there are a lot of clones that will bind to our target.
Right. Also, are sonication protocols similar for radio
yes,
chip and ChIP-Exo?
We use the exact same sonication protocol that we use in our ChIP-Seq, which is 30 seconds on, 30 seconds off for 5 minutes of on time, so 10-minute total cycle with an amplitude of about 42% on our sonicators. Of course, that's going to differ from sonicator to sonicator.
Right. Of course, yeah. So final question here, and then I'll go ahead and wrap it up. So what's the most critical step in your view? Would it be adapter ligation?
In my view, the most critical step for you to, definitely adapter ligation. Without the proper adapters, the whole reaction downstream is stopped. And when we were developing the adapters, that was the problem we kept running into, was that our adapters, if they weren't designed correctly or didn't ligate well, we just didn't get any product to be sequenced basically downstream, and we got results that couldn't really be interpreted.
Right. So that would be a good tip for new users. Make sure that's dialed in. So yeah, as the last question, I'd like to thank once again Bryce Alves for a really insightful webinar and lively discussion, and again to our sponsor Active Motif. We really appreciate everybody taking the time to attend and listen in, and we hope that you've enjoyed this webinar. A recording of the session will be published very soon to the video section on the Epigenie website, and there you can also view other webinars and interviews with thought leaders in epigenetics and non-coding RNA research. So until next time, good luck in your research, and goodbye from everybody here at Epigenie.